welcome everybody. As mentioned, I'll be talking about how I use nanopore technology to resolve complex genomic structures and regulation patterns in cervical cancer. So HPV is necessary but not sufficient in the development of cervical cancer. The normal cervix is made up of epithelial cells that differentiate from a bottom basal layer into terminally differentiated keratinocytes in the upper layers. HPV infects basal stem cells through microabrasions in the outermost part of the cervix called ectocervix. Typically, these infections last 1 to 2 years, but persistence of a latent infection of a high-risk HPV type over several years eventually allows the accumulation of genetic mutations, and this can increase genomic instability and proliferative activities of the cell. And eventually, it can lead to development of high-grade lesions. The HPV genome itself is made up of early genes and late genes, and this describes the point of the viral life cycle that they're expressed. So key genes in the HPV genome include E6 and E7, and these degrade P53 and retinoblastoma, respectively. Integration of the viral genome into the host genome occurs in 70% of HPV-16 infected tumors and all HPV-18 infected tumors. The integrated form of HPV usually removes or truncates E2, and this normally regulates E6 and E7 in a negative feedback loop. And removal of this gene can lead to increased expression of E6 and E7. Integrating HPV also often results in local structural rearrangements because the act of integrating requires the presence of double-stranded breaks. These rearrangements can be simple events, such as the deletion of the human genome during the insertion of the HPV genome, or they can be really complex events with multiple breakpoints in subclonal structures. The example shown here has 2 HPV breakpoints that cause a duplication of the region in between, with the second copy also harboring a deletion within that region. HPV infection and cervical cancer rates have dropped significantly in developed countries thanks to vaccination of oncogenic HPV types, but it remains endemic in low and middle income countries. For example, in sub-Saharan African females, cervical cancer is the second most prevalent cancer type and the #1 cause of cancer-related mortality. So it's still important to study this disease, especially in the populations that are most affected by it. So HPV is often found integrated nearby oncogenes in cervical cancer. In this Circos plot, within our HTMCP cohort, each tick is an integration event, and the ticks are stacked when they are occurring within the same genomic bin as each other. And you can see there are some regions of the genome that are more frequently hit by HPV integration than others. And these often are bins that harbor common oncogenes, such as KLF12, MYC, and TP63. And when HPV integrates nearby these oncogenes, often the integrated sample becomes much more upregulated when compared to the rest of the cohort. So in the box plot, the red dot is the integrated sample, and the gray dots are the rest of the cohort. And you can see for many of these nearby integrated genes, the integrated sample is sometimes several logs higher than the rest of the cohort in expression of the gene. So the learning objectives of this webinar are first to understand how to identify and categorize the virus-mediated structural rearrangements that are often otherwise cryptic to short-read technologies using nanopore technology. And then second, learn how to resolve and visualize complex structural variants with multiple breakpoints. Third, learn how to distinguish and separately analyze the methylation of identical viral segments that are distributed across the tumor genome. And fourth, learn how to use regions that are differentially methylated between haplotypes to identify genomic events like viral integration that affects the epigenome. So this study will be primarily looking at cervical cancers from 2 cohorts. The first is the HIV Tumor Molecular Characterization Project, or HTMCP. And this was published by our group and others in Nature Genetics in 2020. And this has the total cohort is 118 cervical tumors that originated from Uganda, and we have Illumina data for all 118 samples, including whole genome sequencing, RNA sequencing, chip sequencing, and a methylation array. And on 42 of those samples, we also did gDNA and soon-to-be cDNA nanopore sequencing. So our goal is to have whole genome, whole methylome, and whole transcriptome on these. And then the TCGA cohort consists of 192 cervical tumors, and these originate from North America. And they were analyzed in their study using whole genome sequencing, whole exome sequencing, RNA sequencing, and also a DNA methylation array. And on 17 of these samples, we also did gDNA sequencing, so whole genome sequencing. To approach this, we studied the whole genomes using nanopore sequencing. And in our workflow, we extract DNA from the cervical tumor and size select the genomic DNA using blue pippin to get segments of DNA that are greater than 15 kilobases. And these are then sequenced on the PromethION. Our data analysis then consists of base calling and mapping, structural variant and copy number variant calling, de novo assembly, methylation, haplotype phasing, statistical analysis, and and we use the tools referenced and listed here. So our first objective is to identify and categorize virus-mediated structural rearrangements that are otherwise cryptic to short-read technologies using nanopore sequencing. In almost all our samples, it was observed that HPV integrates within human regions that have gained at least 2 double-stranded breaks, while 2 double-stranded breaks also occur in the HPV genome to break it into a linearized segment. We refer to the collection of breakpoints that contain all HPV integration junctions within a linked region as an integration event. To call HPV integration breakpoints, structural variant calling was performed on a hybrid HG38 with HPV genome, reference genome, so that these breakpoints would be detected as translocations. The breakpoints were then grouped into events if one of the 2 conditions were fulfilled. Either the breakpoints co-occur on one or more of the same read, or the breakpoints are within 500 kb of each other. The benefit of long reads is that adding the condition that the breakpoints can be paired if they co-occur on the same read, we can identify integration events that span multiple chromosomes, or are linked together through great distances through structural rearrangements. And without these, we can identify that. Next, the reads with this from each event were subsetted out. So it's just the event reads, and these were used to create local assemblies of the integration events. And using the assembly and read alignment patterns, I was then able to categorize each event as a particular integration type. In certain samples, HPV was more integrated and sometimes hyperintegrated compared to the others. So this plot shows the number of integration events on top per sample. Each sample is one of the columns here. And the number of breakpoints is lists the bar on the bottom. And then other covariants of the sample are also listed, such as HPV type, and the clade that HPV type belongs to, the HIV status in the case of the HTMCP cohort, the histology of the sample, as well as some other molecular characteristics of the tumor. So as a whole, HPV16 seem to be more hyper-integrated than, or more often have more than one integration event than other HPV types. But the most integrated sample was a HPV58 sample that contained 18 integration events comprised of over 80 breakpoints. So using the method I described before, I was able to categorize HPV integration events into 6 categories. And I kind of followed this flowchart shown here. So if the reads had repeats detected on them, I categorized as a repeat integration. If not, I looked at the number of breakpoints. And if there's more than 3, it can either be a multi-breakpoint integration or a complex ECC-DNA integration, ECC-DNA being extrachromosomal circular DNA. If there's 2 breakpoints, I look at the coverage between the breakpoints. So if there is no coverage, it's a deletion. If there is coverage, I move on and see if there's coverage outside the breakpoints. And if there is, it's duplication. And if not, it's an ECC-DNA integration. So here's just a schematic of these integration types I'm describing. Deletion occurs when the B segment that's between the 2 breakpoints becomes deleted when HPV becomes inserted. And in contrast, in a duplication, that B segment then goes in a repeating duplicating structure along with HPV. So you kind of get this A, B, HPV, B, HPV repeat going. The ECC-DNA integration is when that B segment actually gets excised from the chromosome and circularizes and exists as an extrachromosomal circular DNA. Translocation occurs when the breakpoints are on different chromosomes, and this brings the chromosomes together and causes sometimes a full arm translocation event. Multi-breakpoint is when you have more than 2 breakpoints, and this results in several rearrangements of the genome around it. And a repeat is when those integration breakpoints are occurring within a repeated region, and you get an expansion of repeats. So we looked at the number of these integration types across our 57 samples, and the most commonly occurring integration type was the multi-breakpoint event, and this made up about half of the integration events in the cohort. And then the next most common was deletion, followed by ECC-DNA integration. And the rest of them were kind of less commonly occurring. And I also looked at how prevalent these integration types were between the major HPV types. So HPV 16 and 18, most commonly occurring. HPV 45 is the next most commonly occurring. And then I grouped all other HPV types together. So you can see there's some differences here, such as the multi-breakpoint integration is more common in HPV 16 than HPV 18. The duplication integration is more common in HPV 18, and ECC-DNA integration is particularly common in HPV 45 integrated samples. So the 2 breakpoint integration events, the most common are the deletion, the duplication, and the ECC-DNA integration. And this is kind of what the read alignment patterns look like between these 3 types and how I can tell the difference. So in a deletion integration, the 2 HPV breakpoints, denoted here by 2 arrows, and on the bottom in the read alignment by 2 orange lines. These are the regions in which HPV gets integrated between. And you can see in the HPV containing reads that there's a break in coverage of those reads. And this corresponds to a reduction in read depth when looking at the read coverage of the whole genome sequencing. And the region around the deletion can be duplicated. So this kind of happens in conjunction with the event. So the region in between gets deleted, but the surrounding region gets duplicated. And that's what's shown in this example here. In the duplication integration, you can then see the difference where the region in between the two breakpoints has an increase in read coverage in the HPV containing reads, and also read coverage in the whole genome sequencing data. And you can see reads that kind of enter and exit this duplication event. So the reads span the outsides of the breakpoints as well. And this contrasts to the eccDNA integration, where, like the duplication, you have a increase of read coverage between the two breakpoints. But these HPV-containing reads do not appear outside the two breakpoints. So they're contained within it. And that makes sense because you are getting this entire region excised from the genome, and so the reads would not ever move outside these two breakpoints. So we looked at the content of some of these eccDNAs that we found particularly interesting using their assemblies, and 3 are shown here. 2 of these are simple eccDNAs where they're excised from 2 breakpoints from the genome, and 1 is more complex eccDNA, meaning it was rearranged. It has multiple rearranged segments of the human DNA. So the first example on the left shows HPV being nested in an entirely intergenic portion of chromosome 13. And then the one in the middle shows HPV being inserted in a portion of TP63, including 2 exons. And the transcription the direction of transcription is also shown by the arrows. So you can see the TP63 direction of transcription kind of opposes the HPV45 direction of transcription. The complex eccDNA on the right has 3 segments in an intergenic region of chromosome 13 being linked together in the eccDNA in a circle here. And this is along with a segment of HPV52, which becomes inserted as well. So the actual content of the inserted HPV sequence can be variable, as you see in these 3 examples, but all of them show that E67, which are the genes in the pink and the purple, are invariably kept. So these are the minimally required genes in these cancers, it seems. So the second objective is to resolve and visualize complex structural variants using multiple breakpoints. So to do this, I developed a workflow to create a visualization technique to look at these complex structural variants. So first, on the called structural variants across the genome, using sniffles, I subsetted the VCF file to just the associated structural variants. So these are structural variants that share the same reads as the HPV integrations within that event. So yeah, then I get a subset of VCF file with just these associated structural variants. And then I broke the locus into copy number blocks at the structural variant breakpoints. So at each kind of start or end of a structural variant, I broke that into regions in a bed file. So you can see kind of these copy number blocks. And at each separate block, I calculated the read depth within them. So the read coverage I have shown here. And then lastly, I converted this read depth at each copy number block into copy number values. So instead of having a read depth, I then have copy number estimations of each. So here's a simpler example where you can see how I did this. This is the same integration event as the deletion one I showed before. So there's 2 integration breakpoints here along with a duplication event. So it's kind of a deletion insertion nested with a duplication. And this is how it gets rearranged. So the segment in between the 2 gain segments gets deleted, and then there's translocations between chromosome 2, where this is occurring, and HPV 16. And then the region in between gets duplicated in between it. Or sorry, the outside 2 segments gets duplicated in between it. I don't think you can see my cursor, so I'll try to explain the best I can. And then we can apply this to more complicated structural rearrangements and events. So this is a multi-breakpoint event that's on chromosome 8 and has about 5 breakpoints across this region, and has several breakpoints also within the HPV 18 genome here. So you can see the copy number changes quite a bit within the HPV genome. So this gets rearranged as shown here. You can see all of the copy number segments, and there's quite a huge amplification of the HPV genome specifically. It goes up to around 65 copies, and the chromosome, that should be chromosome 8. The chromosome 8 segments gets amplified to about I think 18 or so copy number. So the lines connecting the 2 chromosomes show the translocations, and the blue lines on the HPV 18 genome show the inversions, and the pink line showed duplications. So our third objective is to distinguish and separately analyze the methylation of identical viral segments that are distributed across the tumor genome. So to do this, I'll explain that in those samples where there's more than one integration event, this basically means there's identical, the HPV genome basically has duplicates across the genome. So the sequence is identical. So the reads would pile up on each other. So we have to separate them out in order to look at the methylation of one event versus another event. And since we already subsetted those reads, the HPV containing reads to which integration event they belong to, we can then apply this to look at methylation. So first, determine the integration events and what reads belong to each. And then second, call DNA methylation across the reads containing HPV integration. So each little dash here is a methylation call at a CPG. And then you can separate the reads by integration event and calculate event-specific methylation. So you can, what was all piled together on HPV, and this is kind of what you would see if you had short reads, because you wouldn't be able to separate the reads out. You would just have one overall methylation result for HPV. But then we can separate into events, and you'll see different events have very different methylation patterns. And this would be pretty important when you're trying to understand the biology. So here's the results of the methylation across the HPV genome. And here are different types of events. So I have all the eccDNA events shown together, the deletion events, translocation events, and duplication events. These are all zeroed at the LCR, the position of the HPV, and the dots shown across the row are CPG methylation results. So each dot is a CPG, and when they are blue, they're unmethylated, and when they're red, they're more methylated. So blue unmethylated, red more. And the genic region is then clustered together. So the LCR is the long control region, and this is the non-coding segment of the HPV genome. It's about 500 base pairs, I think, and then it has all the enhancer elements and transcriptional machinery kind of bind in this region. And you can see that it's invariably unmethylated across all the events. So it seems like it's an important regulatory region to keep unmethylated. But the genic region, so all the CPGs that are within these genes after the LCR, can have a bit more variability in their methylation. So eccDNA events seem to have the most consistency. The little triangles here just show the breakpoints where it connects to the human genome. But yeah, the early genic region in the eccDNA events are quite unmethylated, while the later genes in the eccDNA events after that breakpoint tend to be much more methylated. And this is just a very consistent result that we see in these eccDNAs. But there's more variability when we look at the events that are still on the chromosome. So deletion, translocation, duplication shown here. So in the deletion events, some of them also have that early gene demethylation and later gene methylation, but not all of them. Some of them have full methylation of the genic region. And some, like you can see in the duplication events, the HPV 18 containing ones are particularly methylated across the genic region. So I'm trying to investigate the kind of reasons why some would be more methylated, some would be less methylated, kind of within the genic region. But there probably is some sort of position effect variegation going on where the human region around it is affecting the methylation of the HPV genome. Whereas the eccDNA events, you wouldn't have that effect because it's outside the chromosome. So that's kind of my prediction. So moving on to the last objective, using regions that are differentially methylated between haplotypes to identify genomic events, for example, viral integrations that affect the epigenome. So much like what I was explaining with the HPV integration events, the haplotypes often can be confused. It's often important to look at the differences between allelic haplotypes. So whether reads are coming from one allele or another can kind of show you different effects of how they are changing from one another. So normally these reads are all piled together across the region, but you can separate the methylation calls using haplotype phasing. So the SNPs within these reads can be used to separate out these two haplotypes. And then you can look at changes that are occurring between the two and how the alleles might be different in terms of how they are methylated and regulated. So I first separated the methylation calls using haplotype phasing and then called differentially methylated regions between the haplotypes. So these are DMRs. You can see kind of a schematic of a block of DMR beneath the two haplotypes. So this is a region where there's different methylation between the alleles. And then I can see on a broader view where the DMRs are occurring and if there's any sort of major clustering of DMRs across the chromosomes. So this schematic of a chromosome, the black lines are DMRs, and you can see kind of I'm trying to show little loci that have particularly high densities of DMRs, and you can map the DMR density accordingly below it. And using these density values, you can find hotspots DMRs that I call DMR hotspots. So these little blocks of frequently occurring DMRs. So I call DMR hotspots, and then density peaks are higher than a null distribution of those DMRs across the chromosome. So here's an example of this in a sample. So the red line on each chromosome are the DMRs and their density. So they kind of peak and flatten out at different regions. And then the blue line is the null distribution of those. And there's little boxes, I'm not sure if you can see, but kind of little boxes above the peaks. These are the DMR hotspots that are called. So the regions where there's significant increase of DMR density. And I wanted to see if these hotspots DMRs corresponded with HPV integration at all, or when they did. So the dotted line, the vertical dotted lines are the HPV integration events within this sample. So you can see there's one in chromosome 4, but one particular one I wanted to point out was in chromosome 5, where it's right on top of a DMR hotspot. And you can see this is quite a large peak of DMRs. So because HPV is integrated on one haplotype, so it's occurring on, it's a kind of monoallelic event in the genome, we can utilize this differential methylation to kind of see where HPV integration is affecting broadly the methylation of the chromosome. So the average hotspot size of HPV overlapping DMRs was about 5 megabases. So that's actually quite a large area that has changes in methylation because of HPV integration. And you can see this isn't true for all the integration events, particularly in this sample. So there's several vertical dotted lines like chromosome 4, chromosome 8, 13, 12 that don't correspond with the DMR hotspot. So we're also trying to see what makes a DMR overlapping HPV integration event differ from a non-DMR overlapping one. So just another way to visualize this, in the first plot, sort of on the leftmost side, the DMRs are shown and they're centered around HPV integration with 10 megabases on either side. So the red line is HPV integration at 0. And this is just showing kind of the density of DMRs across this region. And you can see that the actual placement of the DMRs can have differences between the integration events. So, for example, you can see quite clearly a focal hotspot around HPV in the third row, so the third integration event listed here. But some integration events have more of a unidirectional effect. So the higher density of DMRs leans more on one side or the other of HPV. But the density is very centered. So there's a little bit of differences between the integration events. And then I also wanted to look at the direction of methylation. So whether the HPV integrated haplotype is becoming more methylated or becoming more unmethylated. So the second plot in E shows the methylation results of those CPGs, or sorry, of the DMRs. So whether it's becoming more methylated in the HPV haplotype, or being shown as red, or less methylated, where it's shown as blue. So we're limited to what we can see in this plot, because we have to, the methylation result has to be within the same phase block. So any adjacent phase block that is surrounding it, we can't really correspond a result to those ones. So they're kind of grayed out here. But the actual phase block that HPV is on is shown with the methylation result of each DMR. So some of the events have more demethylation occurring, but a large portion of them are more methylated. So this is quite interesting, because you can kind of have an argument for both. Demethylation, I think, would be expected because of the nature of the HPV genes being oncogenes, and you would expect in cancer they would be more unmethylated, and so they can be more expressed. But methylation also can be argued because it is a virus, and the typical reaction of the cell to a viral integration is to methylate that region. So lastly, the density of DMRs was compared in the sample with integration versus the samples that don't have integration in that region, to see if these DMR hotspots are occurring particularly in that sample, or if they're kind of just a DMR that it happened to integrate nearby. So for several of these, you can see there's significant results of the red integrated sample having higher density of DMRs within that hotspot. But yeah, some of them is just kind of a hotspot anyways, and then HPV integrates near it. So both results are kind of interesting. So in conclusion, there are 6 main categories of HPV integration, with multi-breakpoint integration being the most commonly occurring. When HPV is integrated near 2 double-stranded breaks, when HPV is integrated between 2 double-stranded breaks, this results in a deletion, duplication, eccDNA, or translocation. And different integration event types have different effects on the local DNA methylation, with eccDNA events having the most consistent methylation patterns. And typically in the HPV genome, the more upstream portion of the genic region is more the upper part is more demethylated, and the downstream part is more methylated. And 4, the HPV integration corresponds with long-range differential methylation between allelic haplotypes in the tumor. So with that, I would like to thank everyone that was involved with this project. So everyone at the GSC, and everyone at the Medical College of Wisconsin, and my lab, and my supervisor, Dr. Marco Mara. And thank you all for listening.